Kind words, nice introduction. I didn't know there are so many flattering synonyms for saying that I'm old. And thanks also for the organizing committee of RegGen for inviting me. It's great. It's a great pleasure to be here. Today, I'm going to talk about something that we have been working part of it for at least 12 years now, and a part of it that is fairly new. And this job is kind of converged together in a very interesting way that I really would like you to be aware of. So the old part actually started with us and several other groups aligning and analyzing human and mouse and then other vertebrate genomes back in the 2003 maybe or something. And then we all found some things that, according to everything that we knew about the conservation and sources of selective pressure, shouldn't be there. So I'm talking about these, we call it, I'll be saying HCNEs a lot, will be these highly conserved noncoding elements that depending on who reported them independently, they were called ultra-conserved elements, ultra-conserved regions, conserved noncoding sequences. It's all basically the same things with slightly different thresholds among groups. And if you can see, these elements along the length of a couple of hundred or more of nucleotides, if you compare human sequence with the other sequences of other vertebrates, you can see that these dots here mean identical sequences as in human, which means you have blocks of 30, 40, 50 or more base pairs that in some of these sequences are conserved all the way from human to fish, I mean, across all vertebrates. And nothing that we know about any functional elements and how they work explains this level of conservation. We didn't know it back when they were discovered 13, 14 years ago. We still don't know it. And I think it's about time that more people start looking at it and find out. So the comparison between a protein coding sequence that encodes a perfectly conserved protein, this is histone H4, and one of these slightly less conserved but still extremely well conserved non-coding sequences tells you the difference. So you really have this three base pair periodicity of anything that's selected for at the protein level. On the other hand, these blocks can sometimes be conserved across 200 base pairs between human and chicken without any differences. So our question was, what was the origin of this selective pressure of highly conserved non-coding elements? And everything that people have tried, sort of, the conclusion was probably not, right? Are the dense arrays of transcription factor binding elements? Probably not. Is there an active removal of mutations, for example, by recombination? Probably not. And one thing that still we have a hope for, especially after discovering what I'm going to show you today, is that maybe they have to do something with chromatin structuring, but again, this is still a pure speculation based on correlation. These elements, I've been showing you individual elements zoomed up at the sequence level, but if you zoom out a lot and see how they are distributed along the chromosome, you can see that they're not randomly distributed. Instead, they tend to form fairly dense cluster around a certain genes. And if you just look at, MACE1 is, homologous transcription factor. DAX1 is a homologous transcription factor. HOXD cluster is a cluster of transcription factor proteins, right? Homologous transcription factor proteins. They are not only transcription factors. The biggest clusters here actually are, but pretty much all of them are known developmental transcription factors. So the genes around which you have these clusters of these elements. And even before their distribution was known genome-wide, people found individual instances of this very highly conserved element. They tested them for enhancer activity because it's a non-coding sequence, it's conserved, so it must be enhancer, right? So, and actually it was. So If you test them in reporter construct for enhancers, 50% of these most highly conserved elements will give you something, some kind of spatiotemporally constrained patterns at 11.5 day mouse embryo. So this is the work of Lempenakil, Axel Wiesel, and the people at Berkeley who have tested now more than 1,000 of these elements. And interestingly, so this is four patterns of 4 elements of around the OTX2 gene and 7 elements around SALL1 gene. And you can see that individually tested, they will give wildly different expression patterns. But for example, in the case of these elements clustering around SALL1 gene, if you superimpose them, if you just stack these patterns on top of each other, you will get something that starts to approximate the actual in situ of the SALL1 gene, okay? And based on this and many other examples in the genome, and also using Tom Becker's enhancer trapping, we came to a very simple model of how these elements work. So you have a cluster of these elements in the genome. You have a gene that integrates the inputs from all these enhancers, and they can be megabase 1 to 3 megabases away in mammalian genomes. The famous Sonic Hedgehog enhancer that Alex Stark showed this morning is one of those. And very often, these genes around this target gene are gene poor, at least for protein coding genes, but sometimes you will have other protein coding genes that will have these elements within their introns or beyond, but themselves will have fairly broad, no specific expression, which means that they will not react to this, array of inputs at all. So we call them bystander genes. Some people object to that, but you know, because these are perfectly fine genes, many of them are housekeeping, very essential genes. But we think they are here as a result of frozen evolutionary accidents, simply because if an element was recruited as an enhancer from an intron of a neighboring gene, then that neighboring gene has to stay in cis to the target gene as long as target gene needs it, right? So how do we call this now 10 years ago, these elements genomic regulatory blocks or GRBs. I'll be saying GRBs a lot in this talk, and I want you to remember, so genomic regulatory block or GRB, by that we mean this arrangement that contains target genes, array of all its regulatory elements, long distance regulatory elements, sometimes other genes that are not regulated, okay? So how do we find these regions for individual genes? One of the simplest ways we implemented also a long time ago in our Ancora browser, we just took the track of individual elements and just took a wide sliding window and reported the density of these elements at different thresholds. And it looks actually quite neat. So, for example, these are 2 genomic clusters that we're talking about 2 5 megabase windows. This is around MACE2 gene, which has the highest number of these conserved elements of all genes in human genome, and also around PAC6 genes. And now, if you zoom out to the entire chromosome, in this case chromosome 3, you will see that if you just limit yourself to fairly distant comparisons, you will get fairly clean peaks of these many highly conserved elements that will inevitably lead you to a developmental gene somewhere around the middle. If you go further, so this is actually comparison of human to mouse, human to chicken, human to frog, and human to zebrafish. And if you go further than that, you will find only a handful in Amphioxus or lamprey. But if you go even further away, you will find nothing. Interestingly, if we just go to Drosophila, which has 0 highly conserved elements of this kind conserved with human, and we compare it with 2 other species of Drosophila, which are actually fairly close, like mammals, you will see that you will have also clustering of these very highly conserved elements. And indeed, you will see that in the middle of these peaks, you will have like a textbook collection of developmental transcription factors as well. Interestingly, so if you remember, I showed you the maze2 locus, which has the highest number of these highly conserved elements across vertebrates. And again, none of these elements is conserved all the way down to Drosophila. But if you just look at which element is conserved between Drosophila and other fly, this is Drosophila pseudoobscura and tsetsefly and housefly. And the biggest cluster that we find that is conserved across flies is the homothorax gene, HTH, which is an ortholog of maze genes, right? Also, in the third lineage, when comparing to the genomes of two sea urchins, we also find the maze gene. In this case, sea urchin has only one maze gene because it didn't have whole gene duplications like vertebrates did. Again, this is still, again, the biggest cluster in the genome. None of them actually share any elements visibly at sequence level. Okay. Based on this, we propose that this gene in the ancestor of all these animals already had an equivalent set of elements, but after the lineage split, you will have a slow turnover of these elements. So the original one will get mutated, the new ones will get recruited, or occasionally the old ones will get lost, and given enough time, you will have no conservation between lineages, but you will have lots of very high conservation within the lineage itself. Okay. So one of the questions was actually when you have these target genes and you have these bystander genes in this arrangement that we call a GRB, a genomic regulatory block, is how, why is it that some respond to this long-range regulation and some don't? And of course, you saw enough reason from Alex this morning to understand why. But back when we analyzed it in 2006, I think, so we took Uwe Older's classification of Drosophila promoters into these 5 categories based on composition of these elements. And so by now, actually, Alex will call this one, and this one developmental ones, and these two housekeeping ones, but we didn't know that. And then we looked at which type is assigned with which is overrepresented in which gene ontology category. So with these two, we found all the functions that have to do with development in more general with multicellular regulation. Okay, so anything that involves multiple cells talking to each other will have these two types of promoters. On the other hand, these two promoters will give you intracellular transport translation, this is replication, and also transcription, meaning that these you will find mostly with housekeeping genes. Well, and the third thing is that with the exception of immune system, which is more complicated, all the other functions that we find associated with data-dependent promoters, we call them textbook promoters because that's what textbooks still tell you what the promoter looks like. What we saw is actually that even though, in star-seq, these and these are practically indistinguishable, these ones, initiator plus DP, will almost never occur together with Tata box. So Tata and DP are very, 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 very rarely occur in Drosophila promoters. So, and indeed, the functions of the genes are very, very different. So we have, on one case, actually really embryonic development and differentiation. And on the other hand, with Tata box promoters, we have genes that are specifically expressed at relatively high levels in terminally differentiated tissues. So, and again, so we are looking at, why they, how they work differently, in the sense because for some reason the star-6 setup doesn't really distinguish them very well. Back then, when we did this analysis, we didn't know actually how to find these three classes in vertebrate promoters. In vertebrate, we knew from 2004 and 5 and Phantom 3 analysis using cage data is that we have this, again, Tata box promoter with differentiated tissue-specific genes that is, it has motif-dependent, sharp transcription initiation, less likely to overlap a CPG island versus this group that is typically Tata-less and has CPG islands and these broad promoters with multiple transcription initiation sites. However, so both the housekeeping and most developmental promoters have this kind of architecture. Okay, so we wanted to see how to split this architecture into two groups to distinguish the developmental regulator genes from the housekeeping ones. And so that was done by Alton-Akalin, also a long time ago, and he showed that practically the biggest feature that distinguishes the sequence feature that distinguishes what we call GRB target genes versus bystander genes is the actual size and number of CPG islands overlapping the gene. These will have huge target genes will have huge CPG islands, sometimes multiple CPG islands running into the body of the gene. On the other hand, these broadly expressed bystander genes will have typically one and only overlapping the promoter. And of course, if you just look at which epigenetic marks are associated with developmental genes, you will find that these will all be padded by HTK4 trimethyl when the gene is active. They will be padded by Polycom and HTK27 trimethyl mark when they're not. And really, if you, in most of these arrangements, the target gene is fairly easy to pick this way. And we have an automated method, now two automated methods to detect these genes. And if you're a developmental biologist, I'll bet you that your favorite gene is probably in this list. So what I have shown you in this case is, this arrangement is devoted to regulating this developmental target gene. And it also, it is in mammals on the order of magnitude half a megabase, 2 megabases, okay? And somehow these regulatory elements have to communicate with this target gene. And one of the questions that we wanted to ask after all the Hi-C papers have appeared is, since Hi-C papers have detected topologically associating domains as one of the fairly stable structures in chromatin conformation, we wanted to see, okay, is there any relationship between this area, these regions that are defined by this clusters of ultra conservation and topologically short-chain domains? Because similarities they are both postulated to be involved in long-range contacts. The size order of magnitude is about right, but differences are, of course, TADs have to be reconstituted after every mitosis exit, right? So after every cell cycle, they have to be reassembled again. On the other hand, and also people who call them, they insisted to segment the entire genome into TADs, more or less. And our genomic regulatory blocks, actually, no, they do not have to be constituted every cell cycle because they have been around for 200, 300, 400 millions of years. Okay, but so and then to do this simple comparison is we just needed to have a good way of finding the spans of these clusters of conservation. And I'm not going to go into technical details, but the method that we have we have shown it's fairly robust both to the choice of species. So you can use human comparison to opossum or chicken with multiple thresholds, or with fish. And most of the big ones, clusters, are fairly stable. And once we have done that, we wanted to now compare it with the actual high-C data to see if there is a correspondence with TADS. So this is like a botched animation, but essentially these three are only three of the 847 of these clusters that we found between human and chicken, for example. And then when we stack them, sort them by size, and just check, so this is the actual density track from which they were derived, and also the individual elements. That's all unsurprising. But now we can, in this heat map, we can visualize anything that, any other piece of data, and which shows a directionality index. For those who don't know, it's a summary of high-C interaction data, which says actually if it's red, then it has a surplus of interactions to the left, to the right. If it's blue, it has a surplus of interactions to the left. So a blue to red transition is the equivalent of a TAD boundary. Okay. So when we visualize this, actually, and on some screens it looks better than the others. You can see it's noisy, but the funnel is there. If there was no relationship, there would be no reason to be there. One interesting thing about it is that people say that these, maybe I'll just show you better in the next slide. So the question is actually, have these TADs been conserved for hundreds of millions of years? And the question is, do they expand and shrink with the genome? And then is extreme noncoding conservation connected to their establishment or maintenance? So the answers to those are actually yes, and we still don't know. But to just show it in a slightly better picture, so one interesting thing about these correspondence of these genomic regulatory block with topologically associated domains is, if the old genome is segmented into TADs, if invariably what you have next to one TAD is another TAD immediately, then you would expect, you have this red directionality index to the left, which should be, and blue to the right, basically defining the boundaries of TADs. But if the next ones are right next to these ones in the funnel, you would expect another red band outside and another on the right, and another blue band on the outside on the left. And we don't see it. So, which means that probably, so our observation is that these TADs are much stronger than anything else called as TADs in the genome. Okay? So, again, unsurprisingly, these structures are stable across different cell types, and they are not reproduced by any of the active or poised enhancer marks. So which means, this is just not just a way to visualize where the enhancers are. And individually, so this is how some of those things look. So these are some of the uncontroversial ones. But for example, in some cases, we know that actually SALL1 and TOX3 are two target genes of these GRBs, but these GRBs are so close together that based on conservation only, you cannot separate them. And now you can just complement this by the TAD. So the GRB model will tell you what the most likely target gene is, and the actual TAD data will tell you when you have accidentally merged two close domains into one. Several other things about these elements is again, as I've told you, these TADs are both stronger and bigger than most of the other TADs. So there are several types of repetitive elements that are actively depleted in those regions, especially science, but several others as well. And so the the CTCF at boundaries looks the way you would expect CTCF at boundaries to look. It's slightly less well-defined at the edge of the GRBs that are called from these highly conserved elements, which is understandable because the boundary is the last element, the outermost elements that detected, right? But interestingly, so this asymmetry is important because it tells you that whenever actually CTCF and the GRB called GRB boundary don't match, the GRB boundary is always inside the TAD and not the outside. Okay, what you would expect these elements to be. So what about Drosophila? Again, I've told you that these clusters are completely unrelated at sequence level. Drosophila genome is much smaller, but again, these clusters occur around the same kinds of genes, the orthologs of genes around which we find them in vertebrates. And when we do the same thing with Drosophila data, this is from Sexton original data, it looks the same, only even cleaner, more beautiful, because at the top you had, in vertebrates, you have quite a lot of probably merged ones, but here everything's pretty much one TAD, okay? And we also compared it to Bas van Stensel's 5 chromatin colors. And we were able to explain, why people find everything, all the housekeeping genes pretty much outside. You have some of them, like occasional bystanders inside, but essentially, so as a postulated regulatory region, these TADs are typically either completely silent or polycomb repressed. You can see quite a lot of blue, and when they're activated, then you have only the gene lighting up as red as regulatory chromatin. Everything that's constitutive euchromatin is at the boundaries, but we argue not at the boundaries, outside the boundaries. Okay, so that just really you will see them at the boundaries because people just didn't orient them, didn't say inside GRB, outside the GRB. They just said actually one on the other side of the TAD, and they called everything as TADs. Okay. So and again, so what I forgot to mention is while in vertebrates I showed you 8 megabase windows for this funnel. In Drosophila, everything fits nicely in a 2 megabase window. So everything's shrunk so that this entire array of conserved elements and developmental enhancers that Drosophila genes, developmental genes receive is actually much more compact in Drosophila. And we can see that in this plot. So when we called GRBs and TADs, when we have the data, you can see that actually everything scales nicely. So one of the things that actually, we had an argument with a difficult referee. If you're in the room, I apologize. Is that okay? But you're just seeing compartments. The people who call TADs also discovered that actually they segregate into A compartment, which has active genes, and B compartment, which has silent or repressed genes. And based on our GRB model, that's not how it should work, because as I've shown you with this GRB, which has a target gene and potentially one or more bystander genes will be always active. Not very highly active, but always active because they are broadly expressed genes. Target gene will not. So the question is actually what happens with the compartment in this case. And we have looked at several loci that we have. So we have this actually 3 different of these TADs that we also called as GRBs. And our presumed target, in this case you have ZEB2, OTX2, and SOX2. And so and these are 5 encoded cell lines and the expression level in them. And what's interesting is that clearly, so you can see that the target genes are the ones that actually can be most highly expressed in some cell lines, or completely silent like SOX2 is in these. Interestingly, so when we look at the target gene and look at where, in which compartment was it actually assigned, so we can see that every time it shoots up, it's in the A compartment. Every time it's completely silent, it's in the B compartment. That's what the TAD people have shown. So this target gene actually behaves, I mean, really its expression level or is predictive of in which compartment you're going to find the whole thing. But actually you will find in most cases the whole thing in the whole region in the same compartment. And indeed, for example, like in these cases, you will have the whole region in the same compartment, but the other genes, which are, we presume these bystander genes, they just don't care. So they can be dragged from A compartment to B compartment. The expression of them will actually hardly change. And probably it's the activity of the target gene that drags them from A compartment to B compartment. But again, in this particular case, you can show that actually there are genes in TADs whose expression is pretty much uninformative for in which compartment you're going to find them. Okay? So one actually short thing is that what could be done with genomic regulatory block model is something that we published it 7 years ago. And that's actually when those first genome-wide association studies for type 2 diabetes and obesity came out. Many of the variants landed in non-coding regions, some of which were very far from the gene. And they were always assigned to the closest gene, in this case, bold gene. And we looked at all these loci, and we said, actually, okay, in some cases, the developmental, they landed in something they would call genomic regulatory block, because it has lots of these highly conserved elements. In some cases, they were assigned to developmental presumed target gene in the region, but in some cases, they were not. And one thing that we argued, it was very controversial back then, actually, that the region in the first intron of FTO, which was the gene that has high association with obesity, as reported, had actually a cluster, they were part of the cluster of these highly conserved elements that most likely, as a target gene, has the IRX3 gene, which is half a megabase away, okay? And so Tom Becker in Bergen in Norway, he tested all these elements in fish, and so this is his data, and we said, actually, these are probably elements that control IRX3, and they are very close or even overlap the actual variant that was detected. And of course, 4 years after that, or 5 years after this, so there are two papers that used the technologies that appeared in the meantime, including 4C, to show that actual elements from the first intron of FTO contact with both IRX3 and even IRX5, which is the next gene. So, and so this is, to conclude with things actually, so genomic regulatory blocks and strongest, the most stable TADs, we think, underlie the same ancient genomic phenomenon. So what this means is that if you have a cluster of conserved elements that corresponds very nicely and predicts very nicely the boundaries of a topologically associated domain, and this cluster consists of elements that have been conserved across 200, 300, 400 millions of years, then the boundaries of TADs are probably the same in the ancestor, governed by the same cluster. So both will scale with genome size and can evolved by genome duplications. I haven't gone too much into detail, but actually it's also a nice story. And so when you unify the two concepts, so you get two benefits. First, genomic regulatory blocks provide a framework for long-range regulation that tell you that if you detected some co-regulation, you really have to still distinguish these genes that we call target genes, which have different promoters and definitely a different level of responsiveness to these elements than what you call bystander genes. And of course, for these regions, topological associative domains will provide more precise boundaries and separation of closed genomic regulatory blocks. Okay, so compartments is actually are predictive of the activity of only these developmental genes and not other genes in the region. Of course, if you do it statistically, there will be correlation between activity of the gene in a TAD and where it which compartment it belongs to, but it doesn't affect all genes, and it's important to it. And then for all developmental regulation and maybe even of these conserved elements in TAD formation, we still just don't know. I will just thank my group, especially these are the people, especially Nathan, who did most of this work. Matthias Meckerschlager was the collaborator. And so I will just leave this model here that summarizes everything. They should be probably out next week in Nature Communications, so you can read it. Thank you.
Questions for Boris?
Have you looked at these in the context of super enhancers, your GRBs? Is there any?
So, okay. Some of those, so super enhancers are typically much shorter than GRBs, okay? So the GRBs span multiple genes, can be 1 to 3 megabases. You don't have super enhancers. And super enhancers, you will find some super enhancers in this structure. But again, I'm not talking about super enhancer concept because if you chop them up like you do all these elements, you will find separate activities within them. Okay, but again, there is no one-to-one correspondence, definitely.
Okay, so let's thank Boris once again and we move on to the next. Oh,
sorry.
I had one question. So, are you arguing that, like, if we have like a GWAS-NIP anywhere on the GRB, there are more chances, let's say, that the target gene of that SNP is the target gene of the GRB?
Yes.
But then surely all of the other bystander genes also have regulatory regions in
yes.
the GRB. So is it like a probability thing? There are more chances that...
So, okay, so of course this model is a simplification. You can have some more complicated scenarios, but typical bystander gene, like when you look at them in any of these loci, is relatively broadly expressed. And the broadly expressed genes, if they have enhancers, and I don't know because most of Alex's housekeeping enhancers actually overlap the promoter. But if they have these enhancers, they are not long-range enhancers. Okay, so if this is sufficiently far away from the what you call bystander gene, I would think the further away it is, the less chance there is to regulate it.
Thanks.
Thank you.
All right, thanks,
All right.
Boris.
Thank you.